to the lens, right? Either flatten the lens or make it rounder, right? So that you can focus either on faraway things or close-up things. Good. So I think that's where we left off, right? We talked all about that. Very good. And we also noticed that ciliary muscle is under parasympathetic stimulation. So that's how we're going to get that muscle to contract is through the parasympathetic system. So far, so good? All right. And did we mention which cranial nerve that was carried in? Did we get that? For specifically getting that ciliary muscle, parasympathetic. I don't know if we actually got to that. Maybe not. So what do you think? Do we remember? I know that goes back to anatomy. Cranial nerves. Huh? Yeah, so there's oculomotor nerve. Which number was ocular? Well, okay, so let's do a little bit of a review. So what was oculomotor nerve was cranial nerve number 3, and then optic nerve was cranial nerve number 2. Okay, so let's stick with 2. What's that in charge of? We just from anatomy, huh? Vision. So like kind of straightforward, getting the light in, right? And we'll talk all about that, right? Turning that light into electrical signals. And cranial nerve number 3, oculomotor, had a few things, right? If we remember, huh? You can move the eye, like with those extraocular muscles. We're going to look at those also in detail today. We can get those to contract and move the eyeball around, some of those muscles anyway. And, there was some other thing. There was a component in that cranial nerve number 3, right? That there's like a parasympathetic component in there, right? So let's see. We have some things, right? We have some things to talk about. So I'm going to actually go to the next couple of slides, and we'll kind of complete this whole picture. So to build the whole picture, right? Let's also talk about a little bit of physics. We've got light traveling through air, and here they're giving us a glass lens, right? And showing us that when light waves hit a lens, right, we change from air to some sort of solid, in this case solid material, and that will cause the light waves to bend. And so they're showing us, right, this bending here. So if we take that whole idea and apply it to the eyeball, we'll have light coming in. They're giving us just 2 points out in space somewhere, and light coming from each point and aiming for that eyeball. And when the light hits the cornea, we're traveling from air to something solid, right? That cornea is solid, right? It's solid tissue. So we are actually going to bend. Remember, the cornea is perfectly clear, right? No blood vessels, perfectly clear, but it is solid, so it is a change from air. So those light rays are going to bend, right? So we see a pretty big bend there in this case. So we'll bend, and then, remember, what is it? Let's review anatomy a little more. What was in this space here? You were good. Okay, very good. Aqueous humor. And then we're going to hit that lens, and then we're going to bend again. It turns out everybody always talks about the lens, because it's kind of like the lens of a camera or something like that, right? And you would think, or maybe the lens of a microscope. Remember we used microscopes in anatomy, and you had to focus, right? So everybody always, kind of thinks about that lens, but it turns out in real life, most of the bending actually happens at the cornea, right? Because that's the first time you switch from air to something solid. So that's where you actually get most of your bending. But there is going to be some additional bending at the lens, right? Depending on what is the shape of the lens, right? Which we started talking about in that previous slide. All right, so we'll bend some more in that lens, depending on the shape. And then we will hopefully focus the rays right on the retina. So we remember the retina, right? They're showing you in yellow. Is here. We'll look at a close-up of that in a little bit. hopefully you're focusing those rays to a point, right? When you hit that retina, and if you can make a point, then you're focused, right? And then we'll talk about what happens over there. All right, so this is our kind of basic, right, intro to that. So let's look at, we've got some object that's far away, right, over here. When something is far away, you can sort of simplify the situation and say that, okay, the light rays that are approaching the eyeball are going to be hitting that cornea sort of parallel, right, kind of parallel to the ground and parallel to each other. I guess that kind of also creates like a kind of a right angle, right, if you're really hitting the center of that cornea. So we're going to hit the cornea with these light rays kind of parallel to the ground because they're coming from far away. And we get to the cornea, we have a bend, right? And then we travel through the lens, hit the lens, we have another bend. And then focus. So this is what we talked about before, right? But now we're specifically saying, okay, we're going to set the object that we're trying to focus on far away. Good, so that the light rays are coming in parallel to the ground. All right, so that would be something far away in focus, right? So it registers by the time it gets to your brain as clear, right? Like it's sharply focused, you can tell what it is easily, right? And it looks good. All right, good. Let's bring this object closer to the eye. So see how they put it closer? All right, when you have something close to the eye, the light rays that come from that object and hit the eye, right, hit that cornea, are approaching the eye at an angle, like a wide angle. So instead of coming parallel, right, to the ground, they're coming at that eye at this sort of wide angle. Good, because we push the object closer. So, and that's just kind of basic physics. So for people who haven't had physics, you're going to have to accept that. That's just the way things work, like light works. So, and they've drawn this for us here, right? You see here, this looks more parallel, and then here, this looks more like an angle. So that's what they're showing us here. So if we're going to hit that cornea at an angle like this at the cornea, then we're going to bend, right? And then bend again at the lens. So we'll bend here, and then we'll have to bend at the lens. Wow, looks like, actually I want to go to this one, then we'll look at that one. Let's look at, oh my goodness, sorry about that, let me turn that up. Good grief. Let's see. Oh wait. Oh wait. There we go. Let's look at this one first, and then I'll backtrack to this one. Here, same close-up object, right? And light rays hitting that cornea at that wide angle. So we bend, and here, this particular lens, see how this lens is the rounder shape compared to these two lenses, which are the flatter shape. So in this situation, the lens has changed its shape, right, to that rounder, fatter, plumper shape. So this lens is able to really bend the rays extra, right, compared to the flatter lens. So if we can bend the rays more, right, we can still get those rays to hit a point on that retina. So this object, when you bring it closer, will still be clear, right? Will still be in focus. That's what's going to register in your brain is a nice crisp image. But here the object is in the same place, so the rays are coming in at that same angle, and the cornea, in this case, is still bending the same, but this lens is flatter than this one. So when the lens is flatter, it's not going to bend the rays as much, right? So if these rays are not being bent as much at the lens as they are here, you see how, oops, the point where we try to focus, it's actually behind the eyeball, right? So that, and it does, like if it's behind the eyeball, that's not going to the brain, is it? Right? Because we don't ever need like neurons behind the eyeball. There's like a pad of fat behind the eyeball. That's useless, right? So at the retina, you see how it's not the point, right? It's It's sort of on its way to the point, but too bad, right? No cigar, right? Just too bad. It's blurry if we don't have a point. So this image is going to register in our brain. We're going to perceive that as a blurry object. It's fuzzy. We can't really see it clearly, right? Makes sense? So this whole process of changing the shape of the lens when we're trying to focus on an object that's close, right? So we go, let's say, from looking at something far away, and now I want to focus on something up close, right? Or if it's like this and this, right? Where I switch from looking out there, and now I want to look over here. Something like that. We'll call that process accommodation. It's on the top there. I apologize, I got it out. But accommodation is this whole process. That's the name. So we're for sure going to know, right? The name and the process. Good? Let's see. Oh, got cut off again. Presbyopia, that's what's on the top there. I should write it. Presbyopia is the name of the problem of not being able to do that, right? Not being able to accommodate properly. So everybody sees it. Presbyopia. Okay. There we go. This particular problem is actually not a problem with the ciliary muscle, right? Which is what we already know is in charge of changing the shape of the lens, right? It's not a problem with the muscle, but it's a problem with the lens itself, right? We lose elasticity in the tissue of the lens as we age, so that no matter what the ciliary muscle tries to do, too bad, right? The lens is not going to become plump very easily. So then you have to wear the reading glasses. Good? All right. Let's see. Looking at the pupil. And remember we have iris, right? Remembering anatomy. Within that iris, which is the colorful, right, part of the eye, we have embedded some smooth muscle, right? And two smooth muscles. One is a sphincter, right? And one is a muscle that has the fibers running radially like the spokes of a bicycle. So the sphincter muscle is able to, when it contracts, make the pupil smaller, right? Which is constriction. And when the radial muscle contracts, it's able to pull that pupil bigger, right? Dilation. So we see here, constriction, here, dilation. Good? So we're definitely going to know this slide, right, and these pictures. And we see here, they make it really obvious for us, parasympathetic stimulation of that sphincter muscle is how you're going to get that constriction. And sympathetic stimulation of that radial muscle is how we're going to open up, right, or dilate that pupil. Good? I think it's very straightforward. Let's see. So getting back to that whole cranial nerve thing, right? Let's come back to that. There's cranial nerve number 3, which was oculomotor. Very good. So oculomotor speaks to the movement of the eyeball, like that name, but we're definitely going to know that there's this other component to oculomotor nerve, which is this parasympathetic component, right? So parasympathetic component, oculomotor nerve, cranial nerve number 3, right? Working this. Good so far? Okay. Let's be sure that we have it really straight in our heads that this sympathetic component, right? The sympathetic nerve that's in charge of dilation of the pupil is not carried in any cranial nerve. So we can make a blanket statement, I know we did it in anatomy, but we'll say it again for sure, that cranial nerves, like none of them have any sympathetic stuff. We can say that, right? No sympathetic stuff. Some of them have parasympathetic stuff, but none of them have sympathetic stuff. Sympathetic kind of works outside of the cranial nerves. we can have stuff happen. Remember, cranial nerves were in charge of a lot of things that happen in the head and neck, except for that vagus nerve, right? Talk all about vagus for the rest of the course, right? Working on hard and all other things. But the rest of the cranial nerves tended to focus on stuff kind of in the head and neck. But we can have parasympathetic in some of those things, but not sympathetic. Sympathetic is going to run outside of the actual cranial nerves. They're kind of on their own. Good? All right. Let's see. Let's look also at the names before we leave this slide. You see meiosis for constriction. Be sure that you don't confuse this word with this word, which is also meiosis. Oh, brother, right? But we have an E in here. What was this meiosis? Yes, cellular, huh? What was it again? Yeah, making like reproductive cells, right? And what was, just to review, what was mitosis with the T? Mitosis. Right, like regular, right? Regular cell reproduction, like all the other cells. All right. So good. So don't get those three words confused.
Sorry, cranial nerve 3. So that has to do with the sphincter muscles and B, and then cranial nerve 2 is I'm just trying to distinguish the two. And then cranial nerve 2 is when the light is entering.
Exactly. So this is going to be kind of our next thing, right? Is figuring out what about cranial nerve number 2, right? That's going to be our next thing. So hang on to that. But let's get these vocabulary words down first. Mydriasis is our other new vocabulary word, and that's just our official word for dilation. Good. So we'll know the official word as well. All right. Very good. Oh my goodness, wait a second. Also, before we leave this slide, let's take note. Whoa. Let's take note under pupil dilation. Like, how are we going to do that? Oh my goodness. Norepinephrine is going to be our neurotransmitter. And we know we use norepinephrine in the sympathetic division, which we've been talking about anyway. Alpha receptors. Whoa. Do we remember that? Right? Alpha receptors. I bet, I don't know if it's coming up, but we will look back if it's not coming up. I bet that's on that big scary table. Remember the scary table? I bet it's on there. Take a look. But I don't know. I don't know if I put it in as a reminder. But anyway, we'll take a look. So far, we've talked about accommodation, right, where we have to change the shape of that lens when we bring something close, and we try to focus on something close. And we've talked about either constricting or dilating the pupil. Let's also take note before we leave this slide that we want constriction of the pupil to happen at the same time as focusing on something close. Does that make sense? Right? We want those things to happen together. So this is why they're both governed, right, in the same system, parasympathetic division. So we can just like, let's do that all at the same time, right? So that's helpful. We're efficient. Good. Let's see. So let's look at, this is normal, right? Normal focusing. We've got a faraway object. We bend, and we focus on the retina. So that's great. Look at the shape of this eyeball here. See how this eyeball is longer than this one? Right? So this is not a normally shaped eyeball. And if everything else remains the same, right? We've got a faraway object. We bend, the lens is working fine, right? Oops, but the eyeball is physically longer. Like, that's just the way it is. It's built in that particular person. Oops, you see how the point of focus falls before you even hit the retina, right? So that, I mean, oops, we didn't hit the retina, we're just going to keep going. And so now the rays actually cross, and by the time they hit the retina, it's not a point. It's like, oops, like spread out. So we know that's not focused if it's not a point, right? So this image is going to be sent to the brain, and we're going to perceive that as blurry. This particular situation happens in a person who's nearsighted or myopic. Good, so those two words are interchangeable. You should know both. All right, so a nearsighted person can easily see stuff that's up close, like without glasses or contacts, but without the glasses or contacts, far away, blurry. Good? And here they're showing you the type of lens that you could use to correct that and get the rays to focus on the retina. So what happens here is this is supposed to represent either glasses or contacts, but let's use glasses because they're kind of easier to talk about. So if we have glasses here, you see the shape of this lens. This is a little bit of physics, but you see the shape of this lens is a concave shape as opposed to a convex shape. So a concave shape will bend the rays in the opposite direction. They'll bend them outward, right? So if you hit if you bend the rays outward, see how they hit that cornea like sort of further apart, and then the cornea will bend the same, and then the lens will bend the same as it always does. But because we started out farther apart, we bring that point of focus onto the retina. Good? All right, so that's that one. And then our opposite problem, I guess there's normal again. And let's look at this eyeball. See how this eyeball is shorter, right, than that one? So we've got, here they're showing us an object that's up close, and those rays hit at kind of a wide angle, and we bend, and oops, the point of focus is behind, right, behind the eyeball. We saw something like that before. And this is going to be blurry. So this situation is the way things are built in a person who is farsighted or hyperopic. So we'll learn those two words. All right. To correct this situation, we use this. You see the convex lens, and that will keep the rays hitting that cornea closer than they were before, so that we bring that point of focus onto the retina.
That's easy to follow when, like, from birth, to understand, like, different shapes of eyes. But what about individuals that, you know, when at the age, all of a sudden somebody has to start wearing glasses? Is that just because of the ciliary muscles, or is their eye also changing shape?
So two different things. A lot of times, you know, a little kid will have normal vision, and then let's say they hit junior high, right? And then suddenly they have to start wearing glasses, right? Could be either, right? Either case. That has to do with built into their DNA. Eventually, one of these shapes is sort of destined to be their result,
Okay.
right?
Okay.
It just hasn't shown up yet, right? But then they grow, right? Everything grows, and eventually, oops, the improperly shaped eyeball sort of develops, appears, right? And so that's one thing that shows up later. I mean, okay, junior high, it's like whatever, 13 years, right, later or whatever, or maybe high school, let's say 16 years later, something like that. The other type of later, which is like middle age, let's say like 45, you're 45 and then you have to wear the reading glasses, that's that other slide with accommodation.
Okay.
And talking about when we mentioned the lens losing its elasticity. So ciliary muscle still works, but the lens, because it's not as elastic, right? You can pull on that ciliary muscle all you want or relax it all you want, right? Too bad. You're pulling on this thing that's kind of stiff, so it's not going to change its shape. Good? All right. Very good. Whatever. It's not that good. But it's good that we understand, I hope. So, let's take a close-up look at this retina. All right, so this is partly anatomy review, but we're going to add a whole bunch of details onto anatomy review. So, this is the side of the retina that's facing the inside of the eyeball, right? And so the lens is over there and the cornea is over there, and the light is coming coming in from over there, and it's hitting the retina here. Good. And the light passes through the retina first and hits that choroid, which is the outside layer of the eyeball. And then, because the choroid has what in it? What pigment? Review from anatomy. What pigment? Huh? Melanin. Ooh. So melanin. Good. Alright, so melanin, right? So melanin in the choroid. Because of that melanin, some rays are going to be absorbed, but then other ones, like if we're looking at something red, right? The wavelengths that are red, that particular red, are going to bounce back, right? Bounce off of that melanin, boom, and right, bounce, and then come back at these rods and cones. So we remember rods and cones from anatomy. So now we're going to look at these rods and cones a little bit more closely. So, you know, become familiar with the shape and know which one is which. I mean, you want to know this slide, right? I put in here to help guide you asterisks next to rod, cone, bipolar cell. You see bipolar cell are these pink ones. They seem to run in the same direction, right? They're like in a series from the rods and cones. And so we're going to communicate, let's say, cone, bipolar cell, and then another asterisk, ganglion cell. It's the ganglion cell. That's why I wrote this bigger. The ganglion cell that actually sends its axon with other axons of other ganglion cells nearby to become that optic nerve, cranial nerve number 2. Good? So this is definitely an important one to know, ganglion cell. Good? Because we bundle these axons to become optic nerve. All right, so far so good. I did not put asterisks next to horizontal cell, which you see that purple, right? That's kind of connecting, you know, various rods and cones, right? Sort of, maybe if they're not next to each other or something, they want to communicate. And also amacrine, which are these orangey cells, kind of connecting some of the ganglion cells. I thought that's getting crazy, right? Let's just remember the ones that are like in this sequence, right? How we're going to go from like a cone, bipolar to a ganglion. That's good. That's good. All right, so the asterisk ones. Let's remember those. Super awesome. Let's see. A little bit of introductory stuff before we get to like the worst slide in the lecture. Okay, go ahead. Wait, let's postpone it a little longer.
But if you're an albino and you have like a melanin deficiency, does that take mess up your sight?
It does, actually. Probably the first thing that happens is that you're super sensitive even to a little bit of light. So that's why people who are albinos oftentimes wear dark glasses even if they're inside. Like you know, sunglasses or tinted glasses.
Okay.
Good?
Yeah.
Yeah. See, stuff gets explained. It's amazing. All right, very good. That's good. So, let's see. Oh yeah, we're going to do a little bit of introductory stuff, like setting ourselves up for a bad slide. I'll admit the one coming up that I know what it is. It's pretty bad. So, but if we can get through that, we're good. Rods. We know we should remember from anatomy because we have just a little touch of it. Rods are going to be sensitive to dim light, so they work really, really well, like in the dark or if it's kind of dim. Cones, we know, are sensitive to bright light, and they give us our color vision. So we've got the wavelengths red, green, and blue, and those are the ones, we'll see a little bit more detail later, those are the ones that are going to trigger, right, the cones, so that we can perceive color. And they work really, really well in bright light. So like this situation, cones, going crazy. All right. Let's establish this. The rods are going to be considered to be off in the bright light, but they'll be considered to be on in dim light. Good? So keep that in mind when we get to the bad slide. And then, ooh, glutamate. Glutamate. We're going to take a look at that. So keep that in your head too. All right, let's see. Let's see. You can look at home, right, as you're studying and stuff, at this YouTube video. It's pretty nice. It's an animation, and it has a little explanation that kind of goes along with it. But I don't know. I think the pictures are nice, and it's nice to sort of just watch, how they did that. So you can take a look at that. Here's phototransduction. We already know the word transduction. So phototransduction is specifically converting light energy to, the electrical energy that we can use, gradient potentials and action potentials. Good. So we've got that. And oh my goodness, this is the super huge, important. I guess that's a paragraph that we want to keep in our heads for the next yucky slide. All right. So let's keep this in our heads. In the dark, right? I like starting the conversation about how do we achieve this phototransduction, like by thinking about, okay, let's start ourselves out in the dark. I think it works out the best that way. So, in the dark, we're going to use, remember GTP, not ATP, right? We've had a discussion before about GTP, right? Very briefly. Very briefly. It was some of that Krebs cycle, right? It's kind of the bottom, right? We had a little discussion about that. And I said, well, we're just going to notice it now. We'll know it's there. We're going to have another discussion about it later. I think this is it. All right? So this is a little bit of a different version of that discussion, right? This is the detailed version of that discussion that we had before. So GTP, similar to ATP, except what are we using? What was the G again? Let's review. Good. Oh, super awesome. There wasn't even a pause there. That was great. So, okay, GTP. And we're going to see that can be converted to something called cyclical. The lowercase c stands for cyclical, and GMP. M is for mono, so 1-phosphate, right? I know we've seen GD, we've seen GT for tri, we've seen D for di, and now we're seeing GM for mono, right? And we put a lowercase c in front of it, so we've got cyclical GM mono phosphate. Good. Cyclical, referring to its shape, right? Like a circular shape. Okay, fine. We don't have to know the details of the shape, right? Chemical formula, but we're going to know C means cyclical, and cyclical means shaped like a circle. Good enough. Great. We're going to see that this cyclical GMP is going to bind itself to a ligand-gated sodium-calcium channel. We'll see it in a second, right? Sodium-calcium channel, ligand-gated. We just picked that out of the pie. I don't know if I have the pie review. We should check back. Because, off the top of my head, I'm not sure if this channel's in the original pie. However, remember I warned you, like as we go through the course, we're going to add a few things to the pie that like the author didn't put in the pie. So that by the time we get to the final exam review, we're going to have a little bit more stuff in our pie, right? So this is one of the things, I'm pretty sure we have to add this one in, right? We'll review for sure. But, all right, sodium calcium channel, ligand gated, because we've got a chemical binding to it. Fine, okay, great. So, that action opens the channel. So far so good, keeps it open. So far so good, keep that in mind, If it stays open, sodium and calcium we're going to see are going to be both coming in together the whole time that it's open. Good? So the key is here, keeping it open. As long as that's bound to that channel, we keep it open, stuff keeps coming in. Good? When stuff keeps coming in, we're going to create a resting membrane potential in rods and cones. This is how we create the resting membrane potential. Remember, all the other cells we've talked about so far, what did we use to create resting membrane potential? What protein? Sodium, potassium, right? ATPase pump, right? So here we see our huge exception to the rule, right? Huge exception to the rule. We see another exception to the rule. Our resting membrane potential, instead of being what?
-55.
Well, resting, that was threshold. 70, right? So -55 was threshold, -70 was resting for most of the neurons, right? That we were going to talk about. Here's our another huge exception to that little rule, right? -35. So let's for sure totally get this, right? We're totally going to remember this thing. Good? All right. Let's see. We're going to do a whole bunch of stuff, right? We're going to look at the next slide to see, like, oh, we do this, we do that, we do this, we do that. Some things, right? And we're going to do some things to, well, cyclical GMP, right? And make it not cyclical. If we make something that is cyclical, not cyclical, right? We go from a circle to not a circle, we've changed its shape, right? If we change the shape of a chemical and that chemical was our ligand, what are we going to do? What's going to happen? And let's say the cyclical ligand was what was bound. We just said that, right? Can't bind, right? So if we change the shape from cyclical to not cyclical, that's regular GMP without the little c in front of it, right? If now it's let's say it's a straight, right? It's not round, it's a straight shape. We change the shape, we can't bind, so it leaves our ion channel. Ooh, what's going to happen if we leave the ion channel? Yeah, right? We change that shape of that protein, and we can close the channel. Oh, so now what's not flowing in? Right, our sodium and calcium, right? So that's going to change stuff, isn't it? Right? It's going to hyperpolarize. Good, become more negative because we don't have all that positive stuff coming in. Make sense? All right, this hyperpolarization from -35 to something more negative, bold, that's your graded potential in a rod or a cone. Good? So it's a little bit of a different mechanism. A lot of the details are different. The overall idea is kind of the same, right? The way we created graded potentials all along was just open up a channel, let some stuff flow, and you can either go, this way or this way, right? EPSP or IPSP. Here in rods and cones, we see, well, it's kind of the same, but we have some details that are different, right? We use cyclical GMP. We can change it to regular GMP. Okay, we didn't use that before. That's new, right? We have a different protein to create the resting membrane potential, right? Before sodium potassium ATP as well. Here, sodium calcium ion channel, ligand gated, right? So some of the details are different. Resting membrane potential, different number, right? We start off a little bit higher, but overall ideas similar enough that you can understand this. Good. All right. So this, wow, if you can wrap your head around this, the picture's going to be not too bad. All right. So let's see.
You're saying that literally in the dark, absent of light, this happens to generate graded potential for both rods and cones?
So let's see. Now we have to work out even more details. Good. This is why I warned you. Like the next thing is pretty bad. All right. So let's see. All right. It's pretty bad. I'll admit this. Truly, this is the worst part of this entire lecture. Like this chapter, this is it. This is the best. So, having said that, you do want to, right? As always, as I always say, just memorize this, right? But let's also, of course, understand what we're memorizing. So, let's see. Here they are specifically showing us a cone, but we're going to sort of understand, right? And then we'll clarify, little details after we get this basic stuff down. The rods and cones basically work the same. It's just when, right? When, under what conditions are they on or off, right? Working the hardest or not really working. sort of the mechanics of how they're going to work are going to be the same. So let's keep in mind, they're showing us, this is from our book, they're showing us a cone here, but we will know, and I wrote it here as somewhere as a reminder, I think over here, we will know that, yeah, the rods are going to have the same mechanics. Good? All this stuff. So let's go ahead and assume, right, or say, okay, they're going to work both in basically the same way. It's just a matter of when, which we already established. Rods work better when it's dim or dark, and the cones are going to be working better, right, or harder, however you want to think of that, bright light. Good? And we can also say about the cones, they'll respond to the color wavelengths also. All right, so let's just look at the mechanics, whether it's rods or cones. Right, so set that aside for a second. Let's just look at the mechanics. So let's start up here. So I tried to kind of put additional stuff compared to what's written in the book and sort of work out the numbering over here. I don't know, to kind of guide you through this particular drawing. So we're going to start up here at 1A, right? This is just where I like to start. I know that that video, that YouTube video that I put up there, they actually start in a different place. I think they start over here and kind of work around, but I like to start up here. I just think it makes more sense. So keep that in mind as well, that different books and different videos will kind of start in different places. But I like to start up here. So let's say it's dark, and we're going to set up a situation, right? We're just going to set everything up, basically that resting membrane potential, right? We're going to set it up, which we just talked about. But let's see, now we can see it like in a picture format. Here is our protein embedded in the membrane. Oops, it's an enzyme. I put it in parentheses because you don't have to memorize the name of the enzyme, but let's just acknowledge it for our discussion, right? So this particular enzyme works on the GTP, right? Just as, remember, we've seen like ATPases, right? A couple different ATPases, they worked on ATP, fine, right? This one is going to work on GTP, and it's going to take that GTP and go ahead and do that conversion that we've already discussed to make it cyclical GMP, the monophosphate shaped like a circle. Great, fine, good enough. All right, this item we already know is our ligand, right? Our circle-shaped ligand that binds to the ion channel that we have chosen from our pie to help us create a resting membrane potential in this membrane. Okay, great. So there's our ion channel, it's closed. We use that cyclical GMP to bind to it. See how it's bound? Channel is open. Because this channel is built specifically for sodium and calcium to travel, both together, go for it, right? So we see this coming in. Great. And that gives us that resting membrane potential of -35. We just put an electrode in, we measure it, -35. Resting membrane potential. Super awesome. Okay. So that's all this stuff. Let's come down to here. Now we're going to change the situation. Now we're going to have some light enter into the situation. Remember here, we had said, let's start off in the dark, right? So now, let's let some light in. Great. Like we were asleep and now we wake up. Good. Dark room, now we go into a light room. Whatever. Okay, so light comes in, and we know that the visible light is all that range of different wavelengths. You don't have to memorize, all the different wavelengths and which color is which, but just know we've got this range of wavelengths that the human eye is sensitive to. So some wavelength is going to enter in depending on what we're looking at and what colors are in that object that we're looking at, and that's going to enter in. And let's see, what is this little complex of proteins also embedded into this membrane, right? Also embedded into the membrane. Let's just remember when we were talking about other neurons in general. And we had said, yeah, we have sodium, potassium, ATPase pump, creating and maintaining our resting membrane potential. That was one type of protein that we had to have embedded in the membrane. We also chose some ion channels if we wanted to create in that location graded potentials. We could also choose if we wanted to create action potentials. If we're at the axon hillock, we would choose. What do we have to choose to create an action potential? We had to choose a pair. What was the pair of ion channels that we had to choose to create action potential? Right, a sodium ion channel and a separate, right, potassium channel. Good, and those were clearly different from the sodium potassium ATPase pump, right? Okay, but if we wanted to create an action potential, we had to have all three of those things embedded in that patch of membrane, right? It was just stuff that we choose. If we want to do something, we have to build it. Good? All right, so here we want to do something, right? We want to take light energy and make graded potentials, right? So far, we've chosen here this ion channel to create the resting membrane potential. We did need a little bit of help from this enzyme, right, to sort of create our ligand. Okay, so we had to choose those. Now we're gonna choose a little bit more, right? We're gonna choose this complex of proteins and also embed those into the membrane. Good, so that we can go from the resting membrane potential that we've created and we're maintaining to, let's go ahead and make that that graded potential, right? Okay, so this is how we build because we want to do this thing. Great. So let's look at the components of this complex, right? Components of this complex. Look at this item first. This item first. Well, let's look at the whole thing. So we've got rhodopsin. So we have to know, now we've got all new vocabulary, all new names of things. Rhodopsin equals this protein that is looking pink in our drawing called opsin, plus retinol. Retinol, some people have heard of this. Retinol, which is represented in our drawing, they drew a little tiny version of the chemical structure of retinol. Good? So we see that here looks like a ring with a carbon chain attached onto it. And this particular carbon chain has a very specific configuration. See how in this drawing they've drawn it so that it bends downward, right? That's significant, that chain is bending downward. Good? All right, hang on. So we have these two items, and they're like a unit, right? We build them together. And we put them together, we have this whole thing rhodopsin. So far, so good? All right. This retinol, this molecule, and it's specifically bent downward so far. Chemical that comes from, vitamin A. Wow. So if it's a vitamin, where can we put that in like our boxes? We have so many boxes in our heads now, right? Vitamins were used as, in the body, used as cofactors, or you could say like coenzymes, right? Yeah. Okay, good, but coenzymes are like a subset of cofactors. So, wow. All right. So far, we've seen what vitamins have we used so far? We had, I think, 3 B vitamins so far. Niacin, riboflavin, and pantothenic. Woo! Super awesome, you guys. That was like a couple weeks ago, right? Or was it only 1 week ago? That's a little scary if it was only 1 week ago. And I'm thinking, that was weeks ago. But anyway, good, right? Super good. Be sure, and this is why I lecture this way, be sure that you're always doing that. Like, vitamins, I remember a little bit about vitamins. What is the other stuff that I remember from before? Because this is the stuff that comes up on the final, right? Like, what are a whole bunch of vitamins, right? that we use as coenzymes in all these different locations. Like that would be a whole series of questions on the final exam, right? Like, ooh, the little coenzyme vitamin section, right? And I'll ask you like 4 or 5 questions on that. All right, so let's see. Wow. So vitamin A, so now we have a new vitamin that we definitely want to put in the head. All right, vitamin A, coenzyme, that happens to have that shape, right? And we attach it to this protein embedded in this cell's membrane, right? And when we put those things together, we just have some names. Good? All right, so just good. Awesome, awesome so far. All right, so now let's go back to, we're going to have the light come in. All right, whew. Light is going to come in and have an effect on that retinol molecule, right? Remember I said that shape was really important, that they're showing us that sort of bent shape. So when the light energy hits that retinol, it changes the shape of the retinol. And what they're showing us here, I suppose, is, I don't know, I guess it's a straight shape, but it'll change its shape, right? And I think in the video, I kind of liked it better in the video because they show you. It's kind of cool because it's an animation. They show you changing the shape, and that looks kind of nice. So anyway, it's gonna change shape. Wow, what happens whenever we change? We know, right? We could guess this now. What's gonna happen if you change the shape of something and it's attached to a protein and blah blah blah? Something's gotta happen, right? Right? Changing shape. What do you think's gonna happen? Huh? Something's gotta happen, huh?
It won't fit.
Won't fit. I like that. Yeah, I like that. Let's see. Let's see some things that are going to happen. So it turns out we have to get this involved, right? Because remember we were over here before this all happened. We were maintaining this resting membrane potential, right? And we had already said that maybe one way to change that was like to actually close that channel, right? And we could get a hyperpolarization. So it turns out we're going to link these two things together, right? Let's go ahead and do that. So to do that, we've got to pull that cyclical GMP off of there, right? To pull that off to close this channel. The best way to pull that off is to change it, right? Change it. Let's convert it. Remember, we said that, actually we said that in the previous slide. Let's convert it from cyclical to, let's say, straight, right? Regular GMP. We've changed the shape. We've pulled it off of the ion channel. We close this, right? But we link this, right, to this. It's this complex that will do that for us, right? Change that shape and get it off of that ion channel. Good? All right.
So that complex is what makes the GMP turn into GMP because
Right.
it changes the shape of that.
Right, exactly. This complex, when retinol changes its shape, that sort of triggers this whole complex, like this whole thing changes its overall shape, right? And this, you see how this, I put these other two items, the green items in parentheses, because these are enzymes that, I don't want you to worry about the names of the enzymes, but let's just look at it. This particular enzyme works on cyclical GMP, right? You see from the name that it'll do that. So, and we see here, you just go with the arrows, that this cyclical GMP will be converted by this enzyme, which is part of this sort of 3-part complex. It'll change it to regular GMP. When it's regular GMP, it comes off of that ion channel. It doesn't fit anymore.
Just kind of repeating.
Yeah.
So that complex is responsible for triggering or manufacturing the enzyme that works on cyclical GMP, which then pulls off the cyclical, which changes the shape of the GMP from the GMP.
But maybe instead of saying that the complex like manufactures the enzyme, it's more like these are already made, they're already embedded, but this change in retinol, the change in the shape, activates. We could say activate. We could say activates the enzyme, right? Activates the enzyme so that it will take that cyclical GMP, change the shape, and yeah, the rest was good. Uh-huh.
Sorry to like repeat what everybody's saying.
That's all right.
I get it.
Okay, seriously, it's totally fine because this is the worst, right? This is the worst.
So we're changing TP, right, to cGMP. It binds, the ligand it binds, channel opens, sodium, calcium flow in. Okay, so that's, we're in the dark at that part. Okay, so all of a sudden someone turns the light on. Okay, and then the light comes in, we change the shape of the retinol. Okay, and then this enzyme over here is going to change the shape of the cGMP to close the channel. To get GMP. Are we good? That's
So you
Good.
have polarization,
Huh?
which leads to hyperpolarization.
Exactly. Because once after we close this, sodium and calcium can't come in anymore. If we don't have that positive stuff pouring in, we become more negative, right? That's our hyperpolarization.
So we need hyperpolarization in order for this whole equation to work.
Exactly. Exactly.
So you need hyperpolarization for light, to be able to see light, and you need the opposite to see the dark.
Sort of. Well, hang on to that. We're almost there. Let's first like be sure that we have this. We turn on that light. We change the shape of retinol that activates this whole complex. This complex is an enzyme whose job is to, when it's activated, change cyclical GMP to regular shaped GMP. When it's regularly shaped, the ion channel closes, positive stuff is not pouring in any longer, hyperpolarization. That's it. Hyperpolarization is still a graded potential, right? It still counts, right? It's just maybe not what counted before, right, in other areas, but here in the eye, in the retina, we've already had a couple of exceptions, right? Why not just make the whole thing a big exception, right? Might as well. Let's just make the eye a giant exception to all the rules that we had, not all the rules, but a lot of the rules that we had set already. Good? Might as well. So we're going to have, in this particular case, right, where we're in the dark, and we flash some light, right, and we're in a cone, hyperpolarization, that is like, oh, cone, right? That is telling us we're looking at something, right? Good? Hyperpolarization. So far, so good? All right. Go ahead.
So you can't close the sodium calcium channel unless you have that complex, right? There's no way to close without the retinol being changed.
That's the key, right? Really, that's the key to like the whole thing. Make sense? So this, we can even take this little, it's not a little discussion, this big discussion, and now make that leap maybe into nutrition, right? And what do they always say about that vitamin D, vitamin A, I'm sorry, vitamin A. Where does that come from? I mean, what is the most popular thing? Food. Carrots, right? What do they say about those carrots and
For
vision?
your eyes.
Oh, right? Don't they always say that?
Yeah.
Huh.
Beautiful night. Beautiful night.
Huh. Interesting, isn't it? Right?
So retinol is a day protein.
It is, isn't it? Oh, well.
I
Wow,
hear my
it's
mother.
interesting. Huh?
I said I hear my mother.
See? Hate it when they're right. Right? You spend so many years, right? Arguing with your mother, and then
Why couldn't you just explain this?
exactly, right? This should come in the mom manual, and then when the kids argue, just kind of, here, look at this, make it into like a flashcard, right? Just look at this thing, right? Poor kids.
Yeah, definitely.
Mom, I'm just 10 years old, right? Stop. So, but then by the time they get here, they'd be like all over me, right? It wouldn't be such a struggle. Wait, someone had a question. Wait, two people, right?
No.
Let's do. Oh no, you're just stretching. Cool. Go ahead.
I was just curious as to, because you said that this process occurs in the cones as well, but I'm confused as to how that is working.
Okay. So yeah. So now that we've got like the mechanics, right? So now let's try and figure out like rods and cones. Good. So like we said, basically the mechanics are going to be the same, right? Like we're going to pick the same proteins, right? Stuff like that. But here they're showing it as a cone, right? So in the dark, a cone is not working, right? We would consider that to be like off. And then we say, oh, we turn on a light, right? Or we're sleeping and then we open our eyes and the room is light, right? Light comes in and we have all this stuff happen and cone is working. Good? So far so good. Now if we have a rod, and now let's do color later. Let's just do light first. So cone and light. Fine. Rod. So when is a rod good for? Dark or dim. Dark or dim. So even in like super dark, right? You'd have to be like, let's say this, in the darkest situation, there's still going to be the slightest bit of light, right? Even if we close our eyes. Like if everybody closed their eyes now because there's light in the room, like do that now and sort of see what you see, right? You're kind of looking at the insides of your eyelids, but light does kind of seep through your eyelids, right? So you still have some kind of sensation, right, of some blobby colors and maybe the background of those blobby colors is maybe reddish or brownish or right, something like that. There's definitely light coming through. So even in the super, super, super darkest, it would have to be like basically like, oh, I just had a thought of I forget this movie where this girl gets like sort of snatched at a gas station and they put her in a coffin basically and bury her. She wakes up. Shoot. Oh,
I didn't even know that one.
huh? Yeah. But anyway, she wakes up in the thing. Like that would be totally dark because you're underground in a box. But otherwise, right? For most of our normal situations, right? That's a movie. Hopefully nobody's in that situation, but that's gross. But hopefully all the rest of normal situations in life, there will at least be a few wavelengths, right? Or maybe photons. I know that's physics, but somehow entering into your eye. So we can almost still assume that our rods will set themselves up, right? Set themselves up and be a little bit triggered, right? But the rods will be sensitive to these tiny bits of light rather than, we're going to see some more slides and we'll build this conversation, right? So the rods, scratch that, the cones are going to really turn on when we've got a whole lot of these photons and or wavelengths, however you want to think about it, hitting those cones. Rods, they'll they'll go through this process even just a few, right? Even just a few photons are going to come at them, right? But the cones, if it's only a few, like they're not going to respond, they're gonna need more coming at them. Does that make sense? Rods are gonna need more coming at them to be working, let's say, at their peak, right? To be fully turned on, to be fully getting, oh, retinol, 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 woo, right? A whole bunch of these, changing shape, activating, doing all this stuff, the conversion, right? We're gonna need kind of like a flood, let's say, right? Or a relative flood of light. Rods don't need so much. When to be fully, right? Fully, activate, go. Make sense? Sort of. Let's look through a couple more slides. Let's see, we're gonna fill it out. Let's fill out this information. All right. So, we've got, let's say, two different situations that we can think about, right? One is when you go from bright light to dark, right? And you know when you're in really bright light and then you go to dark, you got to adjust, right? It's kind of like, oh man, I can't see, right? And we have kind of a similar thing happen when you go from dark and then suddenly you're in like really, really bright light, and also you have it's a little bit different, but it's still that situation of like, oh, right? Oh man, can't see for a few moments. So we have ways that the eye will help us to adjust, but that does take time to adjust, and we know that, right, from experience, that you have to let your eyes adjust to these different situations. So we'll call them adaptation, right? So we've got dark adaptation when we're letting our eyes adjust to the darkness, and we're going to have light adaptation where we're letting our eyes adjust to bright light all of a sudden. It's not so bad if you go from dark to kind of light, this one over here, but we know, right? We know from experience. It's really when you go from like dark and then suddenly, right? That one. That's the spot.
Is that why it like hurts sometimes when you like go outside in like sun and it turns like your eyes kind of hurt?
Kind of, but it's not so much like, yeah, like you get like either the hurting or like the watery eyes. Like that's gonna be additional stuff, right? On top of this, like these adjustments are specifically in rods and cones on that retina. That stuff where like the watery, right, the tears and everything, that's really separate. If we remember back to anatomy, remember we had lacrimal gland, right, and then lacrimal duct, all of that. So that is more a response to be sure that the cornea is going to be bathed in fluid, because, oh my gosh, we just hit all this light. I don't know, maybe that light is going to be really drying or really hot, something like that. So that is a little bit of a separate reaction to light. But they are coordinated. Good? All right, so that's good. That's good. All right, so let's see. Let's do, I guess it doesn't matter. Let's do the top one first. I thought one would be easier than the other. It's still the same. All right, so adjusting to dark. Adjusting to dark. I put in bold here, we're going to focus on those rods, because we already know sort of by definition that the rods are sensitive to, you only need a little bit, right? You only need little bits of light to get those rods to be working fully. All right. So when it was bright, because we're going from bright to dark, right? So let's say, what is a good example? Like, I don't know what's a good example. Like, if you were driving and you didn't have your sunglasses on, right? And it's really bright, and you're kind of doing this, right, that thing. And suddenly you drive into a tunnel, and it's kind of a long, dark tunnel, right? Oh man, right, that situation. And it's, oh man, that's dangerous too. Because suddenly, oh man, I can't see what's in the tunnel, right? Hopefully those other people have their lights on, coming the other way, right? And that's why they always say, turn on your lights in the tunnel. That's why. All right. So like that situation. So we were in this really, really bright light, right? And we didn't have sunglasses on. And now to switch to the tunnel. So while we were in the bright light situation, it's almost like we have to start. Remember the picture from before? We have to start at the bottom of the picture, right? With this complex that had the retinol in it. Good. So if we were in the bright light, the retinol had already changed, right? Long ago, while we were driving, right? It already changed. It already activated the enzymes. We already did that whole thing, right? We were in that situation already. So far, so good. When we switch suddenly into the tunnel, whoa, now we're not in the bright light anymore. Now we don't have all that light coming in and maintaining all those retinol changes, right? All of that. Suddenly, whoops, we just like cut out a whole bunch of that, right? So, wow, if we're not getting all that stimulation anymore, let's go back, right? Retinol, all the retinol, maybe not all of it, but most of it is going to revert back. So we'll go back to this, remember the bent down shape, which was like our starting shape for retinol? So we'll go from, let's say, here to, whoop, a whole bunch of them just go back to normal. If we go back to normal shape, we change all those enzymes that were activated and doing that cyclical GMP conversion, right, to regular GMP. So if we go back to regular shape, we deactivate the enzymes, we stop the conversion, right? And everything is reverting back to our original state, which was in the dark, the top part of the drawing, right? We are making our way back to our setup in the beginning, right? All right, all that conversion back to the original takes time because we have to wait for all these shapes to change, right? It's taking forever. So that's why we have to wait, right? When we're in that tunnel going, oh man, I hope there's not a car coming at me. I hope I didn't cross over that line, right? When are those walls of the tunnel going to come into focus, right? Right? Oh man, no good. That's why they put those, those markers if it's like multiple lanes. Yeah, because those will reflect, right? You should have your lights on, right? And they'll reflect, and you'll at least see those. Good? Because they're brighter. Make sense? All right. So anyway, that process takes time. Gotta wait for all the conversions. It's very complicated. We already, right? We saw that. It takes time. We gotta wait it out. Good? Hang on. Let's look at the opposite. So we're gonna go from dark and then super bright light. Let's look down here. All, right, of our complexes. All of our complexes are going to be like blasted, right? Whether we're in rods or cones, they're going to be blasted with all this light coming in. If we've got a whole lot of light coming in, like let's say we were either dark or even if we were like regular and you go out to super bright, right? That also is hard to see. So we've suddenly flooded all of those complexes with lots of light, like all the retinol that we've got, right, is going to be just changing shape. I mean, some of it's already changed probably, right? But now it's like we're going to be maxed out, right? We've super maxed out. So we're going to be so maxed out if it's really super bright. We're going to be so maxed out that, we're going to get to a point where, like, if anything is going to register at all, we got to adjust to that, right? And again, this is going to take time, right? Because we maxed everything out. And we use the word bleached for when you're just like, that's it, right? We maxed out everything. And everything got a little bit actually overstimulated. And so we have to wait to adjust to this level, right, of light. And somehow get, like, the rod to calm down. Basically, basically you have to wait for the rods to calm down because the rods don't like to work with a lot of light, right? But you just bleach them out, and they're the ones that are actually kind of messing you up, right? Because the cones don't mind all that light, because that's what they like. They like to work in bright light, but the rods don't. So when the rods get flooded with light, they just go berserk, right? And they need to like take the time to. I don't like to work in this situation, right? Just please give me a moment. I'm gonna go over here. I'm gonna have my coffee or something, right? And I'm just gonna go. Good, but that takes time to wait for the rods to get over that shock. I wasn't supposed to. It's kind of like the rods were supposed to not be working, right, in the bright light, and you came over and you woke them up, right, like you were asleep, and someone comes and like wakes you up, like fire, right, fire, and they wake you up, and then you're and you're like all disoriented, and then they're like just joking. It's a false alarm. Why did you do that, right? And you're mad. That's the rod, right? But then you want to go back to sleep because they probably did it like 3 in the morning, right? Like as a joke. And then like they come home and drunk and they're gonna wake you up, right? I mean, we can make a whole story, right? We can make a whole backstory. But then you're like, oh, I hate you, and I want to go back to sleep. But you know, it takes you a while to go back to sleep because you're mad, right? Woke you up. This joke, right? Right? And so it's going to take you time to go back to sleep. So this is like the situation, right? We got to wait for the rods to get over the anger and finally go to sleep, right? Because I did not want to be awakened. This is not my time, right? Good? All right. So I hope that helps, that situation. That little analogy. All right, let's see. So, we're going to add a little more. Go ahead.
The last slide, you said this happens sooner for the guys in the home.
Wait, what do you mean?
On the last slide,
Yeah, let's go. Okay, good.
the second part, this happens sooner for rods and cones.
They become overwhelmed, is that what you mean?
Yeah.
Oh,
They
okay.
get overwhelmed like sooner, right? Than the time. Yeah. But I think I actually really like this analogy of like when you're kind of woken up in the middle of the night because it's kind of like that, you know, and then you're like angry and you have to wait, you know, to like calm down. So anyway, good. So let's look at color. We're almost done. Then we can take a break. Again, you don't have to memorize the different wavelengths for the different colors. This is just, you know, so kind of FYI, just so we know that there are different wavelengths for each color. And let's know that within the category of cones, oh my gosh, I can't believe I said that. Within the category of cones, some cones are more sensitive to, let's say, these blue wavelengths. Different cones, I think they might be right next to it, right next door, will be more sensitive to, let's say, the green ones. And then another set more sensitive to the red ones. And they'll all be mixed in together, right, all next to each other and stuff like that. That's fine. But different ones are going to respond more strongly, right, have bigger hyperpolarizations, right, to the wavelengths that they are built for. Good? Like their retinol is more sensitive to the red or the green or the blue. Good? So if red hits a cone that has retinol that's built to respond, to change its shape to red, boom, that's going to go right away, right? But if the red hits a cone whose retinol is more sensitive to green, kind of, yeah, there's a red in that for you? All right. Make sense? So that's the color stuff. All right. And then this is just kind of a little experiment where you can stare at this. Oh, we could like turn off lights and stuff like that, but I think most people have done this experiment where you stare at this. You can do it now. Stare at this and then switch your focus over to here, and then you can just shout out what happens if you want to do it now. Tell me if it works with all this light going. You guys. Huh? I heard purple.
Yeah.
Yeah. So you see like the complementary color over here, like a circle of complementary color. So this, we're not going to go into all of the details of this, but this has to do with certain cones being more sensitive, right, to one particular color, and then when you stare at that color, right, they all have changed their shape, and then when you switch your focus to a blank area, like those ones are in the process of like, oh no, it's reverting back, and the opposite color will register on the blank area. So that's kind of the sort of basic explanation of that. All right. So let's remember from anatomy and be sure that it's in our heads, even during physiology, this pathway of light coming in and entering into the eye and then hitting the retina and then leaving through the optic nerve. We've got optic chiasm. Some of the nerves will or axons will cross. Some of them will stay on the same side. And then we go, of course, to thalamus. Everything has to synapse in the thalamus first, right, except for smell. And then we swing back to that primary visual cortex back in occipital lobe. So just remember that. Don't worry about the details of all this. This is crazy, right? The other stuff is crazy enough. So don't worry about all this stuff. But let's remember that for sure. All right. Let me see where we are. Because I know this was super a lot. Oh, we're so close. Oh my gosh. Let's just look at this slide and the next slide. And this is just anatomy review, really, actually. So, anatomy review, extraocular muscles, right, to move the eyeball. And this chart, you do not have to memorize. This is like a crazy chart. But we will play with this stuff a little bit in lab. So I thought I should include it. But these letters represent abbreviations for the different extraocular muscles like medial rectus muscle, right? Lateral rectus muscle. And this slide is kind of getting at how those muscles actually work together to give you all the different directions, right, that you can look in. So again, you do not have to memorize this for the exam or for the final. But we're going to play when we get to the lab. So, you know, you can match up stuff, you know, with the lab. Good. So, this is hearing, right? So let's take a little break. Why don't we come back at let's see, because I want to be sure we have time for lab. Let's come back at 9:35, and maybe we'll see. I'll decide. 9:35.